Hello everybody, I think we'd better make a start. Welcome to this final session, which is on... Can everybody hear me? So this is on translational highlights, and we've got 4 exciting research talks coming up on 4 very different imaging modalities. The order of speakers is slightly different to the one in your program. We're going to start with Dr. Tara Spires-Jones. Tara is a reader and a Chancellor's Fellow at the university, and she's an expert in synapses, neurons, and neurodegenerative disease. So I'll hand you over to Tara.
Thank you. Is the microphone working? Does everybody hear me okay? All right, thanks. So I've only got a few minutes, but you're going to see something a little bit different than what I think you would be expecting from this symposium, because we're moving from whole people and whole animals into post-mortem imaging of very tiny structures. But I hope you'll see that it's very relevant and integrates well with a lot of the brain imaging that's done here. So what I'll talk about today is the modality of synapse imaging with array tomography first, and then I'll give you a couple of snapshots of the different kinds of studies we're using this technique for. So first, why synapses? And I assume that there are a lot of neuroscientists or neurologists in the room and probably almost as excited as I am about them, but these are really essential building blocks of learning, memory, and thinking. You have most of the neurons in your brain. You've had them since before you were born. You get a few new ones and you lose a few as you age or drink too much or bang your head. But the synapses are really dynamic. You get new synapses as you learn things. They get stronger and weaker. And they're really fascinating structures that are also central to the pathology of neurodegenerative and neuropsychiatric diseases. So the reason I'm fascinated by them is because synapse loss is the strongest correlate of dementia and Alzheimer's disease, and it's a shared feature across all the neurodegenerative diseases. Because they're so plastic, the damage to synapses is quite reversible. We can recover synaptic number and synaptic function in animal models of disease and potentially in people, and they're very good druggable targets. A lot of the psychiatric drugs that we have at the moment act at the level of synapses or synaptic transmitters. So this is just a nice image of a cartoon of 2 neurons for those of you, anyone who's not a neuroscientist, and they have beautiful processes called dendrites that receive signals and long processes called axons that send signals, and the point where the axon sends the signal to the dendrite, that's this synapse. This is an electron micrograph. And you can see part of my problem in imaging the proteomic composition of synapses in the scale bar here of 1 micron. So these are really tiny structures below the limit of normal light microscopy. So because the synapses are so small, they're on average half a micron in diameter, and I want to look at the protein composition of individual synapses, I had to adapt some novel technology to be able to image proteins on this scale of individual synapses. So I worked with Christina Makeva and Steve Smith from Stanford, and they developed this really ingenious technique called array tomography for use in normal mouse models. So what they did is they combined electron microscopy type tissue processing with immunofluorescence. So we take small blocks of tissue, and I won't go into too much detail because I'm going to show you a movie in a minute, and we make very thin sections, ultra thin 70 nanometer sections, long ribbons of them like you can see here, and that allows us to overcome the Z resolution limit of light microscopy. The XY resolution is actually okay. It's about 250 nanometers is the diffraction limit of light, but in the Z direction, even with our fanciest laser microscopes, we have about a micron resolution. So this technique is a way of overcoming that Z resolution confound. And what I'll do if I can, this is not my computer, but I'm going to try and show you a video of how we do this, because I imagine it's not something you all do on a day-to-day basis. Oh, is it? Oh, it's here. Okay, thank you. And it's already playing. Okay, so this is, we actually come over here. My lab's based in George Square, but whenever someone donates brain tissue to Colin Smith and his colleagues in the brain bank, we come and pick it up, and we pick up samples about this size, tiny little bits of brain from 38 different brain regions important for learning, memory, and that degenerate in various diseases. And then we make them into even smaller chunks that contain in the cortical regions all the way from layer 1 to layer 6, so from the pia to the white matter. And we make these little tiny blocks that we then fix in formaldehyde for a very specific amount of time. We do it for 2 to 3 hours, and paraformaldehyde there, shaking there. And then they go through steps of dehydration and into a plastic resin called LR white. And this is very much like what you would do for electron microscopy. And then this plastic resin that's originally liquid is baked, so it cures overnight. And the reason we go to all this trouble to bake and cure these samples is because then we can use a diamond knife, shown here. Sorry, that's my 6-year-old at the back. You're using school's out today. Here's the diamond knife, and the diamond knife allows us to cut these 70 nanometer sections. So we get these long ribbons. So that's why we have to have really hard plastic. We can't cut sections that thin with normal techniques. So here is some brain sections coming off onto the diamond knife, and they float on water. And then we collect them with that 6-year-old's eyelash, actually. He's got great eyelashes. We manipulate the brain sections onto a cover slip here, and then we can stain them with normal immunofluorescence techniques. So it's very fiddly and very fine, but it gives us this beautiful high-resolution imaging. So now we're just staining them with, that's just saline, so we stain them with antibodies to synaptic proteins and antibodies to pathological proteins, and we can strip and reprobe these kind of like a Western blot. So we can put on one set of markers, take the images. This is my PhD student, Rosie, who made this movie. She's the talent with the iPhone. So it's taking an image now. Our computer is taking an image of the whole ribbon at low magnification at 10x. And we have some clever macros that kind of do it for us. So this is all these thin sections we were just cutting. You can now see them appearing on the screen here. This is a slow part, so Rosie sped all this up. In fact, this whole 3-minute video is about a week's worth of work. So you're getting a week in the life of a lab person, of a neuroscientist. So here she's got the image of the whole ribbon, and then we zoom up and we find this is a plaque that you see in Alzheimer's disease. So this is an amyloid plaque. So we find an area of interest, and we find it on this big long ribbon, and tell the computer we want to take a picture here. And we click on one of these nuclei in one section, and then the serial next door section, and then the computer can use the nuclear pattern to find that same pattern all the way along the ribbon, and it takes images in the same spot for us all the way along. So we generate a 3-dimensional stack. And now it's going along taking the images in the different channels. You can see the plaque is the big blob, the synapses are the tiny dots, and the nuclei that we use just for the pattern are the sort of bigger round circles. So this is how we collect the raw data at 63x. And then the lovely students and postdocs get to take these data and process them. We have to line them up because they're not perfectly aligned, so I'm sure that you proper imagers are really, really familiar with alignment. So we align them, and then we use some processing techniques to exclude objects in single sections only, because those are usually secondary antibody noise. We're still using threshold-based detection. That's the whole movie, sorry, I'll stop it there. And we've been trying to get away from threshold-based detection methods, but it works pretty well. And what we end up with are, let's see if I can get back to it. I've saved it as a show so the Mac issues wouldn't, there we go. So what we end up with are images in 3 dimensions of really small stuff. And we have a bank of these. So we're taking tissue from every person who donates their brain to Colin Smith's Edinburgh Brain Bank. So if you're interested in looking post-mortem at really small stuff like synapses or mitochondria or other subcellular compartments or little tiny bits of microvasculature, we probably have some cases you'd be quite interested in. We have a lot of Alzheimer's cases. We're collecting the Lothian birth cohort as people donate their brains. We have a lot of ALS cases with cognitive testing that I'll show you in a few minutes. So keep that in mind if you want to collaborate. So now I'll give you a snapshot of a couple of projects so you can see the kind of data we collect. And just as a tiny bit of background, we work mainly on Alzheimer's disease and other degenerative diseases in the lab. Alzheimer's has 3 cardinal pathologies: atrophy, that you can see illustrated here, and the accumulation of plaques and tangles. Plaques are extracellular A-beta deposits, and tangles are intracellular tau deposits, both of which can now be imaged using PET imaging. Plaques better so far than tangles, but it's coming along. And what we think is happening in Alzheimer's disease is that age and other risk factors like the ApoE4 gene lead to an increase in amyloid beta production or failure to clear it. That leads to tau pathology, and this leads to the neuron and synapse loss that subserves the dementia symptoms. So this is called the amyloid cascade hypothesis. But there are a lot of holes in this hypothesis, and so far we are miserably failing to translate this hypothesis into effective therapeutics. So what we're working on is some specific aspects, and in particular, how A-beta and tau are interacting in synapse degeneration. Because of all the things that go wrong in the brains of Alzheimer's patients, the loss of synapses correlates most closely with the dementia symptoms. And we have a lot of evidence indicating this isn't just a downstream effect of neuron loss, but it's actually an early pathogenic feature. And also that synapses are the point where the pathological proteins are spreading through the neural circuits. So we're looking at this using this high resolution technique. And the first question we asked is whether that plaque peptide A-beta is present at synapses in the human brain, where in mice we can show it causing the synaptic damage. And my PhD student, MD PhD student at the time, Robert Kofi, did this when we were still in Harvard. And he found, this is a presynapse in green, a postsynapse in red, so you can see the lovely resolution you get. You get that snowmen, we call them, this apposition that we can see in the images. And the blue stuff is A-beta, and so here you can see a presynapse that has a little bit of A-beta co-localizing with it, and here a postsynapse with some A-beta in it. And what Robert observed was very similar to what we'd seen in a lot of our mouse models. There's an increase in the number of synapses that contain this toxic A-beta as we get close to plaques, and that coincides with a loss of the synapses. So this soluble A-beta is quite synaptotoxic when it hits the synapses. And Robert also saw that people who have a copy of the ApoE4 gene, which is a risk factor, have 5-fold more A-beta at their ApoE-containing synapses than people who don't have ApoE4. So this is really quite interesting to us, and we're following it up now in further studies. And this is just to show you that in collaboration with Tom Gillingwater and Colin Smith, we're also able to do transmission electron microscopy in human tissue now, which, according to the dogma, you just can't do. But Tom and Colin got it working beautifully. So this is in an Alzheimer's brain, an amyloid plaque. This is a big dystrophic swollen neurite around the plaque, and if we zoom in, here's a nice preserved synapse near the plaque. So we're doing a lot of very fine ultrastructural studies in addition to the protein studies we do with array tomography. I'll skip the data, I only have 10 minutes. And then another little sort of snapshot of data is we're working with Sharon Abrahams to look at synapse loss in ALS patients and whether this correlates with their cognitive status. And this paper is out for review right now, so fingers crossed, please. We have some data from Tom's lab showing they found synapse loss in the frontal cortex of ALS patients compared to controls with EM. And they saw an increase in the profiles of degenerating or dark synapses by EM. And this is very labor intensive. If anyone has tried transmission electron microscopy, these studies take a very long time. So we came along and replicated this in about a year with array tomography in 27 cases and 17 controls. And we still see synapse density with maintained cortical thickness. What we also see is that this synapse density correlates a little bit with TDP pathology. And we have a paper that is a collaborating labs paper from Rosa Palicelli and Laurie Rajendran that's come out yesterday in Neuron, showing that TDP43 pathology makes your microglia angry and causes them to engulf synapses. So this is really quite fascinating. And we saw in our human cases that this TDP, sorry, I'm going, oh no. I've got this as a show, and I've never done that before. So yeah, in our human cases, the TDP43 associated with loss of synapses and also cognitive status. If you had cognitive impairment, that associated with synapse loss. So where were we? Here we go. So what we're doing in the lab is looking in the imaging space, at least. We're looking at people's cognition. We get these data from our psychology colleagues. We have great collaborations with Joanna Wardlaw and Mark Baston to be able to use the in vivo MR imaging, particularly of the Lothian Broth cohort subjects. Colin Smith works with us to look at the pathology. We're extending this really deep phenotyping of these cohorts out to the level of synaptic protein and synaptic ultrastructure with these two techniques. And I think this is really important and a powerful set of collaborations that are fairly unique to Edinburgh, so I hope you can take advantage of this. And we're using mouse models and cell models to establish causality and look at disease mechanisms. So imaging is only sort of one side of what we do. Before I say thank you to everyone who did the work, please do get in touch if you have any questions in the future, and we're recruiting, so tell your friends how lovely Edinburgh is. The UK Dementia Research Institute has some positions coming up, and I'm also a member of an early career researchers network that provides some advice, so you could send this on to any early career colleagues for how to set up your lab, how to get money, that sort of thing. So finally, we have some great people in the lab and some fantastic collaborators around Edinburgh, and thank you for having me this afternoon.
Thanks very much, Tara. We started a couple of minutes late, so I think if that's okay,
Yeah,
we'll move on
fine.
to our next speaker. So many of you will know or have worked with Dr. Tom MacGillivray. He's an expert in retinal imaging, and when he's not doing that, he's managing the image analysis core downstairs in Crick. So Tom's going to talk to us about retinal imaging on the cusp of vascular and neurological diseases.
Thanks, Michael. Everyone hear me okay? So thank you very much for the introduction. And I wanted to start just with some headlines. You might have seen some of these exact ones. You might have seen some similar ones. It's the type of headline where the retina and retinal research makes the news. Things like Parkinson's, a test for Parkinson's, Google. I've hit the press a couple of times in recent years. One of the speakers this morning highlighted the Daily Express and their love of a headline. This is one of our studies, eye test to beat heart disease. That wasn't quite how I explained it to the journalist, but that's the headline they wrote. And a Scottish team develops an Alzheimer's eye test. Again, this is another one of our ones. The project is about vascular dementia, but don't let the facts get in the way of a good story. So what I'd like to do is really take you behind these headlines and explain about retinal imaging research, but place that in the context of the programme of research that we do here in Edinburgh. And in the beginning, my contact with this area of this field and this area of imaging started with an image. This is a fundus camera image. Some of you might be quite familiar with this if you go to the opticians and have an eye test, an eye checkup. And of course there was a professor. This is Professor Dillon, who's the clinical chair of ophthalmology. And in about 2004, Bal came to see me with a clinical research fellow called Niall Patton, and the conversation went along the lines of, I've got these images, we'd like to analyse them, we know what we want to measure, but we don't have any software. And that's how my contact with these images started. And at the time, I was working in the clinical research facility, where I'm still closely affiliated, and we'd set up an image analysis core in the CRF. And one of the remits of the image analysis core was to do this very thing, was to help people who had image data but didn't necessarily have any way of analysing that, and to work up new and novel means of extracting measurements from images. And also at the time there were two studies going through the CRF that the CRF was helping to facilitate that were featuring retinal imaging. One was Ian Deary's LBC 1936, the Lothian birth cohort, and the other one was the Edinburgh mild stroke study, which is with Joanna Wardlaw and Fergus Dubel. And they were actually conducting retinal imaging in one of the rooms in the CRF. And Bal was the clinical co-applicant on those projects, and was the person tasked with interpreting that retinal data. And my expertise was very much around extracting measurements from images. And I built up expertise and skills around image processing, looking at the different colour channels that make up a colour image, image processing, filtering operations to enhance the blood vessels. We were very interested in measuring things to do with blood vessels, and then applying things like machine learning techniques to classify where those blood vessels were, but also to separate out things into arteries and veins, so we could distinguish between arteries and veins in these images. So we could go from our fundus camera image to something where we could quantify things to do with blood vessels. And we were interested in the diameters of these blood vessels. We were interested in tortuosity, which is how much they twist and turn. We were interested in the branching pattern, and using techniques like fractal analysis, where you can put a number to the complexity or sparsity of the vascular network that you see there. And we're really interested to look where there might be subtle abnormalities or subtle deviations away from an optimal arrangement of blood vessels that would give us some interesting information. Which then leads on to why the eye, why study the eye. Some of you might have seen this image before when I've used this type of slide. I think it sort of illustrates quite clearly that you can see blood vessels at the back of the eye. It's just about the only place in the human body where you can take a picture and see small blood vessels. And small blood vessels are a really interesting thing to study in the context of small vessel disease in the brain, but also systemic disease elsewhere in the body. But there's not just that, in that the eye and the brain go hand in hand, I'm sure many of you know. They share a blood supply, but they also share nerve tissue. So the retinal ganglion cell layer, the axons of that cellular layer, go through the retinal nerve fibre layer, through the optic nerve head and back to the visual processing and cognitive centres of the brain. So there's a really good rationale there for why studying things to do with the eye and blood vessels and getting an idea of nerve tissue and linking that to things to do with vascular disease and neurological disease. And these are some of the original results we got from those two studies. So Fergus, who was leading on the retinal imaging and looking at that in the context of lacunar stroke, the majority of lacunar stroke is thought to be caused by small disease of the small blood vessels in the brain, so we were using the measurements from retinal vessels, and we saw a difference in veins, in the small veins, the venules between the lacunar stroke patients and the cortical stroke patients who are being used as a control, and we also saw a sparser vascular network in the lacunar stroke group. So we were seeing differences between the lacunar stroke patients and the control group that we had. We didn't see too much in the LBC study. This was about 10 years ago when the LBC participants were about 70. They're quite a healthy bunch. There wasn't really too much wrong with them. We didn't really find too much in terms of when we tried to look at retinal parameters and the cognitive data. but we'll come back to these studies a bit later on. But what these studies did, in conjunction with other studies, was provided the impetus for us to look at developing our expertise around analysing retinal images. And the Vampire collaboration was born. There's another professor here called Manuel Chuco from Dundee. We were working on very similar problems, very similar image processing challenges, and we realised that combining our efforts, we could solve much more collectively than we could do working apart. It was round about the time, or just after Synapse had been formed in the Scottish Imaging Network, and we borrowed that idea. Let's work together, let's work across institutions. And two of the aims there were about establishing a world-class Scottish grouper centre for retinal image analysis research, and we were very mindful about integrating the computational and the clinical side of that. And then also about producing software, so software that would enable studies involving retinal biomarkers by allowing efficient and accurate measurements of the retina. And over the years, nearly 10 years, we've seen about 10 PhD students, past and present, come through our group, and been about 7 postdoc positions, past and present, and I think probably in excess of £2 million in research funding. But it's not all about the research. Of course, there's nights out, team building activities. The picture was back when probably I had a little bit more hair than I do now. But the fundus camera image is not the only way to image the retina. We are, by a quirk of fortune, we have a major technology company on our doorstep. Optos are a manufacturer of retinal imaging devices, and they're just across the water in Fife. And they use a slightly different means of imaging the retina. They're able to access more of the retina with their imaging using a scanning laser approach and mirrors, and able to reach parts of the retina that are inaccessible to a fundus camera. Now, there are challenges in dealing with these images. They capture much more of the spherical surface at the back of the eye, and Optos spent a lot of time looking at how they could deal with this, and the mathematics in the process are quite similar to how you take the globe and unwrap that into a map without distorting structural features. So if you imagine that challenge of unwrapping a globe and not distorting Greenland or not distorting Africa, how do you maintain the consistency and the accuracy of measurements of distance and area? And then, having interacted with Optos in their process of doing that, we were then able to take what we know about doing with fundus camera images and apply that to Optos images, and get to the stage that we could analyse things to do with vessels in these types of images and apply our knowledge and expertise to that. And that was largely thanks to 2 PhD students, Gavin Robertson and Enrico Pellegrini. And Gavin and Enrico came and did postdoc positions with us after their PhDs and are now working for Optos in their R&D department. And Emma Peed is a PhD student who's continuing this work on currently. And there was funding there from Synapse in conjunction with Optos. One of the things that we've been looking at doing, and really the next phase of our collaboration with Optos, is looking at the classification of hypertension using these types of images, these types of ultra-wide field images. So some pilot work that Gavin was looking at was this idea of measuring retinal vessel parameters and looking at whether there were things related to hypertension in there, and could we classify people into hypertensives and non-hypertensives using this retinal data. This is unpublished data at the moment, it's in preparation. But we've got a reasonably good classifier of hypertension. You see the arrow, the ROC curve analysis there. The closer that is to the top left-hand corner, the better the classifier is. But at the time we were using blood pressure measurements from the conventional cuff arrangement, where, as many of you know, there are some limitations with blood pressure taken in that way. It's maybe not the most accurate way to measure blood pressure. So the next phase of our project is to house an Optos machine down at the hypertension clinic at the Western General, and to compare our measurements against a better reference standard, the ambulatory measurements, where someone takes a device home and it monitors their blood pressure over a 24-hour period. And we're due to start this project later this year. So another method of imaging the retina is optical coherence tomography. This gives us a cross-sectional image through the retina, so we can start to look at structure at the different cellular layers at the back of the eye. The most common measurement is to look at retinal nerve fibre layer, so between the red and the blue lines there at the top. and measuring that thickness and the change in that thickness over time gives us an indication of axonal loss. And we know that axonal loss happens in Alzheimer's, in the preceding stage of mild cognitive impairment, also happens in MS. But looking at thicker structures in there, taking the ganglion cell complex layer, which factors in another couple of layers, we can do further mapping and look at further neuronal loss. So in the bottom graph, for the bottom left-hand graph, we have the retinal nerve fiber layer around the optic nerve heads. Then we can move to a more inclusive mapping of that, and then the GCC layer beside that. So we get some quantitation of thinning of the retina that is a marker of neurodegeneration. And this OCT imaging happens in the rolling clinic, so just a couple of 100 yards away from here, where there's an OCT device and people going through the clinical research facility there. And there are 2 PhD students who have been working on this in Edinburgh. James Cameron, who some of you may know, has been using OCT predominantly in MS, but has also been imaging people with early onset dementia and also Parkinson's. And he's collected over 2,000 participant scans and is very close to writing up his PhD. He's very close to finishing his PhD. So there's going to be some exciting data coming out over the next 6 to 12 months. And then Tom Pearson was utilising the Optos scanning laser ophthalmoscope and OCT with a focus on MS and also healthy volunteers. And he's in the midst of data analysis and has also started writing up. So I think coming soon will be some interesting results. And anybody that volunteered for Tom as a healthy volunteer, thank you for that. So just getting towards the end of the slides, OCT angiography is an emerging technology. We have this technology downstairs in the imaging centre where we can look at smaller blood vessels than we would be able to see in the fundus imaging. It's virtual angiography. It's about deciphering the parts of the OCT signal that are scattered, and what's scattering the OCT signal is blood, and we can build up pictures of small blood vessels. And to give you an idea of where this might be of use, this is an N of 2, so it's a very early stage results, but just to sort of highlight the quantification we can do around vessel density and look to see where there's maybe a sparser vascular pattern occurring in the eye and how that might be related to small vessel disease. And in this particular example, in a hemorrhagic stroke patient from the Latch cohort. And again, retinal imaging may be delivering some quantitative measurements about small vessel disease. Where we're bringing all this technology together is in the PREVENT study run by the Centre for Dementia Prevention, where we know there's thinning of the retinal nerve fiber layer in Alzheimer's, there are vascular changes reported in fundus imaging, there are deposits reported in the periphery of the retina, and we're trying to bring all this technology together at an early stage and follow people up. So people are in midlife, potentially at risk of dementia later in life, and see if we can identify where some of these retinal signs, retinal changes might be happening and correlate those with abnormalities as measured on MRI. How am I doing for time, Michael? Okay. So just quickly, the last couple of slides. Going back to LBC and the Edinburgh Mild Stroke Study, we've got lots of data there that we are looking to interpret in a statistical machine learning approach. We have 3 postdocs working on this. We're looking at retinal imaging, signs of small vessel disease from MRI. We've also got clinical data, cognitive data, and record linkage, and looking at employing some advanced statistical techniques, structural equation modelling and machine learning to try and sort out the links between the retina and the brain and see if we can identify some signs of vascular failure in the brain and utilise the data that we acquired many years ago. And then just to summarise some of the future and some of the challenges, measurements, we can always do a little bit better with our measurements, where they're better summative measurements of the state of health of the retina, but also looking at targeted measurements. Maybe we need to target specific areas or specific vessels and increase reliability and reduce uncertainty in those. Multimodality, in terms of the emerging retinal technologies, but also looking at the retina in context of data from MR and PET. Artificial intelligence through the application of machine learning, things like deep learning to data analysis, but also image processing problems. And then some recent developments. There's a regenerative ophthalmology lab that's going to be created from a £1 million charitable donation, which will help us translate some of these advances into better imaging techniques and into studying eye disorders that are a result of neurodegeneration and the new eye eye hospital is planned for the bioquarter in the coming years. Again, an opportunity for us to translate things that we're doing in the imaging centre and on campus here into the patient population. So, thank you very much.
Thank you, Tom. I think we have time for one question. If anybody has a question. Lucy?
Thank you. Hi, Tom. Could you just talk about what the hypothesis is with the hypertension? So why would you want to measure that from your retinal image rather than just with the normal cuff? Is it like a screening?
Yes, yeah, I think it's a sort of people who don't know they've got hypertension. So pretty much everybody goes to the opticians for an eye check, especially as you get a little bit older and people's eyesight starts to change. So I think it's sort of catching people who have hypertension that don't know they've got it. So there's obviously a bit of health economics there to show that that would be worthwhile, rather than dumping a whole new batch of people into the NHS, is actually to show the benefits of catching people early on. And there are benefits to catching people early with hypertension in terms of reducing the number of strokes and heart attacks. But yeah, that's the thinking behind it, is can we discover those undiscovered people who are out there. So the first stage of that is to take that to the hypertension clinic where we know we've definitely got hypertensives.
Let's thank Tom again for his presentation. Okay, so our next speaker is Dr. Fiona Denison, who's a senior lecturer and also an honorary consultant in maternal and fetal medicine. And she's going to tell us about MRI of the healthy and unhealthy pregnant mother.
Thank you very much, Michael, and thank you very much for asking me to speak at this imaging opening symposium. So what I'm going to do for the next 10 minutes or so is just give you a flavour of some of the work that I've been doing with a team of individuals, many of whom in the audience, looking at MRI for pregnancy and seeing whether it has a potential utility or not in looking at the fetus. So before I go on to that, I'm just going to do a wee bit of history in that partly because Scotland was one of the leaders of developing ultrasound in its earlier years. And this is in Glasgow, imaging one of the first pregnant people that they ever did. And as many of you probably know, it was based on ultrasounding and detecting shoals of fish. And that really was in 1960, it was 1958-60, so about 60 or 70 years ago. And ultrasound, as many of you know, has come on leaps and leaps and bounds. And this is a vast difference in images that we now see when we're imaging pregnant women by ultrasound. We get huge detail in fetal anatomy. We get details in fetal blood flow. That's the blood flow through the umbilical cord. And again, although it's slightly trendy and potentially doesn't actually add much to our clinical acumen, there's these 3D images that women want to get to show what their babies look like. So I guess why am I showing you this? I suppose the obvious question is, well, so why do we need MRI if ultrasound is as good as that for imaging pregnancy? We know ultrasound is safe in pregnancy, women like it, it's low cost, etc., and it is readily deliverable across lots and lots of different settings. Well, the challenge is that ultrasound, certainly in the context of pregnancy, delineates structure, doesn't really tell you a huge amount about function. Some of the newer ultrasound methods do, but not really to the level that we want. There are certainly limitations of ultrasound in particular conditions, and the main condition that is really problematic ultrasound is in looking at the placenta, because ultrasound gives you some detail in placenta, but currently we have no test telling us whether a placenta is or is not healthy. And despite the advantages in ultrasound, there's no sign that really ultrasound is potentially going to be able to offer us that. So I think there are some need for new technologies in vivo, particularly for assessment of placental function and fetal well-being in utero, and also potential for biomarker development, because there's an increasing appreciation that actually the health of the fetus in utero sets us up for a long life trajectory. I know you've heard a bit about that earlier on. And potentially if we can actually identify the at-risk and high-risk fetus before it's even been born, first of all there's a chance of intervening when there's maximum plasticity and the developmental structures, and secondly you can actually give some potentially protective therapies round about delivery or in the early neonatal period. So what I'm going to do in the next 10 minutes or so is just give you a summary of some of the things that we've been doing with MRI. And first of all, just to show you about the practicalities, this is a pregnant woman. She's not particularly pregnant, but unlike imaging non-pregnant people, we have to lie women on the left lateral side so we don't sort of crush the inferior vena cava, because the babies don't like that, and it's generally not very good for them. So it does actually pose us some logistical challenges, actually fitting pregnant women in the scanner. And some of the earlier studies we did were on very, very severely obese pregnant women, and I can see Annette smiling in the audience. It was slightly challenging getting some of these very larger ladies in the scanner. But if you are inventive, it's certainly women like being scanned, and actually many of them actually fell asleep, which was not really what we were expecting. So one of the main things that we've been trying to identify, use MRI for, is to see whether it has any utility to identify what I've put as a healthy small or an unhealthy small. And what do I mean by that is ultrasound is good at looking at baby size, but it's not really any good at telling us, or it has limited utility of telling us whether a baby that is small is a healthy baby and potentially is achieving its growth trajectory in utero and therefore can stay till the end of pregnancy, or a baby that's potentially failing to meet its growth potential, either because it itself is sick or the placenta is failing. And the problem that we have as doctors is, do we deliver babies that are sick before they die in utero, or do we keep them inside for longer? So that's what we were trying to use MRI for. So the technique that we've primarily been focusing on, and I'm certainly going to cover in the next wee bit, is spectroscopy. And I don't need to tell the audience about the potential challenges and benefits of spectroscopy, but certainly uniquely for pregnancy and looking at the placenta and brain function, it has the potential for non-invasively assessing metabolism, which ultrasound cannot. And certainly some of the other things that you would use in the non-pregnant situation, such as using gadolinium and other traces, we can't use in pregnancy. So we set out thinking, well, actually, can we use MR spectroscopy to look at the placenta? Well, that hadn't been done before, and could we actually tell anything about the placentas in babies that were well or not well? So what we did is we started off really looking at really sick babies and really healthy babies. And of those was a very small study. We recruited 3 healthy pregnant women and 3 babies that we knew were sick. And this was a very extreme phenotype. We had one severely growth-restricted baby that was discharged in home and oxygen, which is a marker of very poor long-term outcome. We had one baby that was, again, very small and died shortly after delivery, and one baby that died prior to delivery. So these were babies that we knew by our conventional techniques were in trouble. And we did some MR spectroscopy using proton spectroscopy, looking at the placenta. And again, N of 3, so the numbers were small, but we could see lipid peaks in both the placentas. But in our healthy babies, there was a nice big choline peak. In all 3 of our severely compromised pregnancies with knackered placentas, we couldn't detect a choline peak. And when we looked at the lipid to integral choline ratio, there was a sort of significant difference between the MR signature in these N of 3 in different groups. So that was promising, but it wasn't really telling us anything more than we knew already. We knew these babies would be sick, we knew the placenta was sick. So the next step was this: could we potentially use MRS for placental health in a less severe clinical phenotype? In other words, babies that were small, but were not by our conventional techniques compromised. And again, this is where team working is really important. Working with the physicists, we kind of thought, well, actually, what other metabolites can we detect using MR in the placenta? And one of the things that came up that we thought we could try and look at is the glutamine glutamate. And that was interesting, first of all, because we could detect it, because there are a huge number of things that you can detect. And secondly, there are a reasonable number of studies suggesting that actually the glutamate metabolism within the placenta and within the fetus is a kind of potential biomarker of placental health, or could be. So what we did in this study is we recruited 9 matched pairs. So these are 9 women with well-grown babies by ultrasound, and we gestationally matched them to babies that we knew were small by ultrasound. So they were ultrasound, and the abdominal circumference was under the 10th centile. But by all other ultrasound parameters were entirely healthy. For example, they had normal Leica volumes, the blood flowed through the cord, etc., were all fine. So these were not babies that we thought were at risk. We weren't planning on delivering them. And this just shows you an example of spectra. In the solid bar is of our control, and the kind of dotted bars are our SGA, small for gestational age. And we looked at a range of readouts. We looked at choline, we looked at lipid, we looked at glutamate, glutamine, and the vagaries of presenting MR data is that different journals want different ways of the data being presented. And this journal was very adamant they wanted it presented as a water peak as opposed to a ratio. And these are our 9 matched pairs. And again, there was a tantalizing hint that actually in our SGA group, our small babies, the ratios were lower generally, but not exclusively, than our healthy controls. And the 2 that were starred here were the contrary, the ratios were slightly higher. So again, not conclusive, but again suggestive that in these group of, I suppose, cases and controls, there were signals that the placental metabolism potentially was different between the 2 groups, which has been shown ex vivo but not in vivo before, and the MR has a potential utility of picking this up. But to take this forward, what we've really come to appreciate over the last few years is actually the technique is great and it's good, but it needs a lot of work, particularly for this pregnant population, which is challenging compared to the non-pregnant population, with the difficulties in challenging. So what we're doing currently is almost taking a step backwards, but actually it's not really, it's a step backwards to enable us to go forward more robustly. And actually finding, well, actually, what is the normal range? Because nobody knows what the normal range are for these metabolites throughout gestation in a healthy pregnancy. So we need to know that before we can actually realistically plan future larger studies looking at an unhealthy cohort compared to a healthy cohort. And we've just finished recruiting 80 healthy pregnant women from 20 weeks up to 41 weeks gestation. And we've prioritised getting MR spectroscopy data from these, from the placenta from these women compared to other. And we're in the process of analysing this, and this is going to be presented at IFPA, which is an international placental conference by David Morris, one of the physicists in Manchester, at the end of August. And this is just showing you some of the raw data for the lactate signals across gestation on the x-axis. And the other thing that we're doing is actually working with the animal facility and also with our colleagues down at IGMM, is we're actually trying to take placenta and actually say, well, is there anything more that we know that we can get out of this placenta spectroscopy using a higher strength magnet and comparing that to the actual metabolic profiles to see whether actually we can, if there is something else that's coming out, can we redesign or can we make our signal acquisition more intelligent to maximise the data collection and the robustness of it. So the second thing I've said I touched on briefly is can in utero MRI be used as a biomarker for later onset disease, because pregnancy is a window of opportunity for potential intervention. And I'm just going to show you one study that we've been doing with our neonatal, with James Boardman, who's one of our neonatal colleagues who has a very active programme of work really looking at neonatal MR and following growth restricted babies postnatally. And this was looking at a group of women who had diabetes during pregnancy compared to those who had not diabetes during pregnancy. And it was really a proof of concept study looking at whether the feasibility, first of all, recruiting these women, and also was it possible to do a comprehensive assessment of the fetal brain in utero. Now, fetuses are quite difficult to scan because they move. So you can put your voxel in the right place and then you scan it and then they kind of move. So it's just actually quite challenging to get multiple different types of data set from the same women within the same fetus within a dedicated time frame. And the reason why we chose diabetes is because there's emerging epidemiological evidence that actually babies who are born to women who have diabetes during pregnancy are at increased risk of having cognitive problems in later life. In childhood, for example, difficulty concentrating in school and failure to achieve milestones compared to those women who do not have diabetes. So we set out to see whether we could look at fetal brain metabolism. Again, this is in the fetal brain, not placenta. This is show how the voxel localization, looking at brain microstructure using DWI, and again macrostructure using structural MR. And this has just been published quite recently in the American Journal of Neuroradiology. And I'm just going to show you one of the results, which essentially, again, focusing on the spectroscopy, looking at NAA to choline, NAA to creatine, and choline to creatine ratios in the fetal brains in women who've got diabetes compared to healthy controls. And what there is a suggestion that in the brains of the fetuses, there is a difference or a lower choline to creatine ratio compared to women who are healthy controls. And that is potentially biologically plausible, because if you actually look at adults with type 2 diabetes, they have done lots of studies looking at spectroscopy in the adult brain, and actually the one that comes up as potentially being different in the adult brain in type 2 diabetics is the choline to creatine ratio. So potentially there might be something in utero happening in these fetuses that could signal for a later onset development of diabetes. It's difficult to say. So I think what we aim to do in the study, and what we demonstrated, is that it's challenging, but it is possible to get multimodal MR imaging of the fetal brain at 3 Tesla in a single imaging session. It's fair to say that we got better data acquisition of the spectroscopy, and then it tailed off because we ran out of time. And again, that's something that's important when you're planning fetal imaging studies. And although our study was not actually powered to detect definitive differences, in utero MRI has potential to act as a biomarker for later onset disease, and potentially and also to look at results of treatment in pregnancy. So I think that is quite exciting. So taking you back to the beginning, this is what's happened in ultrasound for the evaluation of the healthy and unhealthy pregnant woman. This is what's happened in 60 years. If we've got this kind of scatter thing, which I, to be honest, couldn't tell you exactly what it is. And that's what we now have with our fetal image, with ultrasound imaging. And this is currently where we are, but I don't know where we're going to be in 60 to 70 years. Some people, the argument that people often threw at you when you're talking about MRI in pregnancy is, well, it's never going to be the tool of choice because our health services isn't set up like that. Well, ultrasound was never really the tool of choice, and if you demonstrate the evidence that it changes and is a better modality, then potentially MRI could be not necessarily mainstream, but for those really high-risk pregnancies for which we have nothing else to offer, in the future our service could be potentially designed differently. And that's all I have to say. This is just, I apologise to anybody that I've not put on this slide, but there's a huge big range of individuals, from the physics to clinicians to the midwives, and actually to women and their babies, that have actually volunteered with great gusto to come into the scan. It's one of the few studies I've ever done that you put a poster up and women actually contact you, because normally you have to hunt for them, but women are actually very, very keen to MRI scans. Thank you very much.
Thanks, Fiona. I think, again, we have time for one question.
Oh, one at the back.
Emilio?
Great. Sorry, I came in quite late on that. But I remember writing a paper with Alston McCulloch a number of years ago about choline creatinine ratios. And when we looked at not the ratio, but the absolute numbers, it wasn't the choline that was changing, it was creatinine. Did you have a chance to look at that?
We have done that in our cohort of our small babies where the numbers were small. There was no difference in the absolute choline and the creatine to water. And we haven't done that. We're in the process of looking at the choline creatine ratios with gestation during pregnancy. So I can't tell you, first of all, whether it changes with gestation or not. And I think we need to know that consistently first before we do any further studies.
Thanks again, Fiona.
Thank you.
So our final research presentation of the afternoon is from Professor Kev Dhaliwal. He's Chair of Molecular Imaging and Healthcare Technology and a consultant in respiratory medicine. He's going to tell us about optical imaging with molecular probes.
Thank you very much for the opportunity to be here.
Okay, let's thank Kev and all of the speakers so far in this session. So before we move to our final speaker, just a reminder that there is a reception at 4:30, so it's nearly wine time. So our last speaker of the session is Professor Jonathan Seckel, who's here in, I guess, two capacities, as Professor of Molecular Medicine and also as a Vice Principal of the University. And he's going to draw this session to an end with some closing remarks.
Well, good afternoon, everyone. putting in what I think is the largest grant I've ever written. Probably the largest one ever written by the university. It was all of 9 figures. So we'll see how that goes. I think the chances of getting that sort of fee is pretty minimal. But we will see. We will see. It's gone. So I'm delighted to be here. You've obviously had an absolutely brilliant day. I'm looking through at the titles and what you've been talking about. It's absolutely spectacular. This is one of the jewels in the crown of the university, certainly of the college, with the ability to do just the last thing we heard of from Kev, the ability to actually see in real time molecular events happening in a human, in vivo, in a lung. Extraordinary. It's incredible how imaging has come on. So well done. You're in a jewel in the crown within a college that's a jewel in the crown. I've just got the latest research grant income data. It's going to be a good year, this one, on top of a series of good years. We're doing really well. You've, as a college, you've doubled your grant income. You're already the biggest grant earner in the university. You've doubled it. And you've gone from 92 to 188 million, which, given that the university brings in about 300 million a year, is considerably more than half. And you've got another 2 months to go before the year is finished. So this is a fantastic college doing fantastic things, and imaging right at the heart of it. It's been a great year. We've been getting money from everywhere, doubled the amount of money coming in from research councils. Despite Brexit, anybody in favour of Brexit? That's a real relief. Despite Brexit, we've got 22% more money from the EU. And in fact, I was over talking to people in the EU a couple of weeks ago about maintaining access as best we can. Not clear how that will happen for all of us, but even the UK government, with its tin ears and cloth heads, seems to recognise that we need to do something to keep research, which of course is international, going. So hopefully we'll be able to keep that going. Industry income is up, and charity income is up by about a third. So fantastic. Great unit in the middle of a great college doing wonderful things. We're in a campus where huge amounts are being invested. We've got a brand new neuroscience hospital, only a year behind. It's dear old NHS procurement. That's the way those things go. Brand new neuroscience and paediatric hospital going up. Lots of new buildings. Institute of Regeneration and Repair starting to up the build soon. So lots of exciting activities going here. And imaging? For imaging, most of this would be rather boring and rather dull. So you do lots of wonderful things. And I'm very proud of it. This is a great group. When the notion came to build CRIC and to start trying to coalesce imaging, it was difficult to see whether it was really going to succeed or not. But my has it succeeded. What a fantastic series of capabilities, from imaging fetuses in the human womb through to, Kev's told you, through to the entire gamut of this great day. So well done, well done. I think that's all I need to say, really. You're a great bunch, you're doing some wonderful things, and I'm keeping you from cheese and wine. So well done all day, keep up the good work, and let's commercialise everything that Kev has told us about. Well done, thank you very much.